Thanks for coming today. I'm Ted Price. I'm a professor in behavioral and brain sciences and the director of the undergraduate neuroscience program. I'm glad to see so many people here. Hi, Mom. Hi, Lab. So this is the Brain Matters seminar series. We do this once a year. Greg Dussor and I are the speakers this year. We are going to be talking about work that is going on in our laboratories about pain, and specifically sex differences in pain. So this is a hot topic right now in the pain research field. I think it's a hot topic in neuroscience in general. So sex differences in the nervous system. So for you guys that came here today, I'm sorry, we're going to be almost entirely actually focused on how we're learning about differences in the nervous system that affect females. So hopefully that's still going to be very interesting to everybody. So this is the fourth year that we've done this. Greg and I are really happy to be able to do it. Greg is right over there. And Michael Burton, who's right here, is going to be our moderator. So when we get to the question time after each talk, if you have questions, just raise your hand. Michael will come to you with the microphone. We are recording, so please wait till the microphone comes to you to ask your question. Also, please stick around afterwards because we have food and drinks, and we can mix and mingle and talk about the research that we talked about today. All right, so I'm now going to introduce Greg Dussor. Greg is right over there. Greg is an associate professor in behavioral and brain sciences. Greg did his undergraduate work at the University of Alabama. He then went to the University of Texas Health Science Center in San Antonio, where he did his PhD. Greg and I actually did our PhDs at the same institution in the same lab. We've worked together for almost 20 years now. So we have a long history of collaboration. So after he did his PhD, he went to the Oregon Health Science Center, Health and Sciences Center, or the Volum Institute in Portland, Oregon, to do a postdoctoral fellowship with Ed McCleskey. And then his first faculty position was at the University of Arizona School of Medicine, Department of Pharmacology. And then about 4 years ago, he moved here to UTD. So Greg is focused on pain research, just like I am, but Greg's work really focuses on migraine headache. So he's one of the world's leading experts in migraine headache, mechanisms of migraine headache, and has played a huge role in developing preclinical or animal models of migraine headache, which is a very difficult thing to do, which has had a really gigantic impact on our ability to develop new migraine drugs. So Greg, welcome.
So I'll have to introduce Ted shortly, and a lot of that's going to sound familiar because Ted and I have worked together for 20 years, so a lot of what we've done has been in parallel, so sorry if I repeat. But so as Ted mentioned, I'm going to talk about migraine and sex differences in mechanisms, some of the mechanisms that we're looking at, and what that means for new therapeutics. So first of all, let me just get some disclosures out of the way because we're supposed to do this. I am co-founder of Therapeutics. I'm co-founder of a company that is named after him, which we can talk about later. I get funding from Alder Biopharmaceuticals, and I get consulting fees from these companies, and you can decide for yourself whether any of that matters. Okay, so let me just start off with some numbers. So this is from a publication that came out in 2017, last year, and it was done by the World Health Organization. And what they do is they look at 328 diseases across the entire Earth, and they rank them in terms of how disabling they are. So what are the top disabling diseases across the Earth? And here are the numbers. So number 1, low back pain, and number 2 is migraine. So the most disabling disease on Earth in the year 2016, at least, and it was also that way in 2006, and it was also that way in 1996, is low back pain. And number 2 is migraine. That number has been consistent for many years as well. So 2 of the top 5 most disabling disorders on Earth are pain. Here's more pain, neck pain, musculoskeletal disorders, a lot of that is pain. This is a big deal, very disabling conditions. So those of you who have migraine, I don't have to go through the time course here of how an attack looks, but I'll do that briefly anyway for those of you who are not familiar. There's a period of time when patients are normal, and hopefully that period of time is long. Something triggers an attack. The most commonly reported trigger is stress. It can be fatigue, not sleeping enough, sleeping too much maybe, skipping meals, scents, light, hormones, all kinds of things can be triggers. And that puts patients into what's referred to as a prodrome phase, where any number of things can happen in the prodrome phase. Energy, mood changes. They often know that the migraine is coming. And then about 30% of patients get what is referred to as an aura. This is typically visual disturbances in the visual field. You can have blind spots. You can have geometric patterns that move. That only lasts about an hour at the most. And then you transition into really the disabling part of the disorder, which is the headache phase. And this, by definition, has to last at least 4 hours, but it can go up to 72 hours. Many patients, it goes much longer than that. It's unilateral, throbbing head pain, typically pretty high up on the pain scale. Along with it, you get nausea and vomiting. You can get sensitivity to light and sound and touch, a lot of different things. This is really what keeps people away from work and doing the things that they would otherwise be doing. They want to be in a quiet, dark room and just ride out the pain until it's over. After the headache phase is a postdrome, where patients just don't feel right, and it may take them 24 hours to just get back to normal. So this whole thing can last many days, in some cases a week. This is part of why it's the second most disabling disease on Earth. The other reason why it's the second most disabling disease on Earth is that we're not actually doing a very good job of treating it with currently available drugs. So patients are left really to deal with this without any help. So the other thing that the World Health Organization did in this study was they grouped all the 328 disorders and injuries into categories and colored them. And so here is the disability scale going left for females and going right for males. And bottom to top is age. So this is 0 to 6 days, 28 to 364 days, and then 1 year all the way up to 95 years. And so whether you're female or male, there's not a whole lot of disability going on in your first couple of years of life, but as you age, disability goes up. And you can see that this is pretty nicely skewed in the female direction. And if you really look closely at this, and we don't have time to do this, and you probably can't read all these anyway, there are different disabilities in females than there are in males. And so we're going to talk about migraine. This is, at this point, considered a neurological disorder, so it's this light purple. There's a whole lot of light purple here across age in females, not much of that in males. There's other, this is musculoskeletal disease, very common and disabling in females, not much of that in males. What's disabling males? Cardiovascular disease, respiratory diseases, urogenital disorders, a whole bunch of this green stuff. Guys get in car accidents. Guys do harm to themselves by doing crazy things. So there's more green on the right. You probably expected that was going to happen anyway. But clearly it's skewed in the leftward direction in terms of disabling disorders. And in females and males, the disability is coming from different things. So we're talking specifically about migraine. And here is the commonly reported statistic for migraine. This is age and the prevalence across age. You can see in males, it's pretty flat across age. But in females, 2 to 3 times more common than it is in males. And it's clearly more common during peak reproductive ages. This is the disability, how disabled women in pink and men in blue are across age by migraine. And it's more disabling in women. So it's pretty widely recognized that migraine is both more common and more disabling in women than it is in men. There is clearly some role for hormones in migraine in women, and we don't really understand exactly what that role is. So this is days of the menstrual cycle, this is weeks across pregnancy, and this is age around menopause. And each of these lines is a different hormone. There's, 5 different hormones plotted up there, and clearly they're going up and they're going down, and they go up and down during pregnancy, and they go down during menopause. And what's in gray here is the migraine incidence. And so you can see there's clearly specific times of the cycle when migraines are more likely to happen. They disappear in many women during pregnancy, but they come back right around birth, and certainly in the postpartum period of time, they come back. And then they peak right around menopause as well. So there's something going on with hormones, and it's not something that we understand. What complicates things is that a lot of the hormones are on their downslope when the susceptibility period is, and that's just confusing. So there's been a whole lot of work that's been done on estrogen and progesterone and the usual hormones that you would think of when you look at something like this. And it's just not really clear exactly what they're doing. So we wanted to go down a different road and look at a different hormone, and so we started asking questions about the hormone prolactin. And there are a number of publications that have implicated prolactin in headaches. There are certain kinds of tumors that you can get that cause excessive secretion of prolactin. Those patients often get headaches, and migraines get worse in those patients, and you can treat them and their headaches go away. As many of you know, again, stress is the most commonly reported trigger for migraine, and we know that causes prolactin release from the pituitary gland. We think that stress can cause prolactin release from immune cells, that it can cause prolactin release from blood vessels, and all of this can act on receptors for prolactin that are on nerves that may contribute to pain in women, but maybe not in men. So we ask this question at the top of the slide on a daily basis. What is the source of migraine pain? Where does it actually come from? The brain itself is not sensitive, so you can cut it, you can burn it, you can electrically stimulate it, you can do whatever you want to the brain once it's exposed, and you have no anatomy that allows you to feel your brain. But the meninges that cover the brain are incredibly sensitive to stimulation, and the only sensation that you feel when you stimulate the meninges is pain. These are studies that go back to 1940 in conscious humans. When the meninges were stimulated, those patients reported headache, and you can get patients to report headaches that are very similar to migraines if you stimulate the right location on the meninges. So we think this is the source of the pain, at least the pain phase of migraine. So what we wanted to do was look at the expression of the receptor for prolactin in females versus males. And this, I'm going to transition into talking about mice and rats now. This is somewhat of a genetic trick, but we've introduced a red fluorescent tag onto neurons in the meninges and across the body of these animals that express the prolactin receptor. So anywhere you see red would be a nerve fiber or a cell that has the receptor for prolactin. And so you can see a lot of nerve fibers here, you can see a lot of nerve fibers here. You're looking at the meninges of a female mouse. Doesn't really get interesting until you look at the male. And there's almost no nerve fibers that are red at all. And so basically what this means is that the prolactin receptor is expressed on nerve fibers in female meninges, but not in male meninges. And that's interesting. So if we look at the electrical activity in these neurons that are signaling information from the meninges about pain, and probably the neurons that are responsible for headaches, we look at their electrical activity, and each one of these spikes is an electrical impulse. If you apply prolactin to these neurons, you get more electrical activity. And if you do this in females, here's before prolactin, here's after, you get a really nice increase in the electrical activity. If you do the same thing in males, nothing happens. It's as if they don't respond to prolactin at all. So they don't have the receptor in the meninges on the nerve fibers. They don't respond to prolactin. Those are two interesting things. We can take the meninges out of these animals and look at the release of a specific protein called CGRP. I'm going to come back to that shortly. But if we incubate the female meninges with prolactin, we get more CGRP release. And if we do the same thing in males, it doesn't work. So we're building an argument here that prolactin is doing something in females, and it just doesn't seem to do the same thing in males. And then here's an experiment where we apply prolactin to the meninges of mice, and we watch their behavior. And what you're looking at here on the y-axis is the sensitivity of the skin. And we use this as a measure because many migraine patients, 60 to 80% of migraine patients, during the headache phase of their attacks, they get very hypersensitive skin. They don't want to touch their face, they don't want to touch their scalp, they don't want to brush their hair. That's very irritating. And that's something that we can measure in these animals. Obviously, we can't talk to these mice, and we can't ask them, are you having a migraine, and what does it feel like? But we can interpret from their behavior that they're having a headache because they display behaviors that humans also display. So a drop When this blue line goes down, it basically means that those animals had hypersensitive skin in response to the prolactin. And you can see that this is females in blue. Very nice hypersensitivity response in the females, but we do the exact same thing to the male, nothing happens. It's as if we didn't do anything to them. They just don't seem to respond. So all these sort of fit together and suggest that prolactin is doing something in the context of headache on these nerve fibers that are signaling from the meninges, and it's only doing it in females. Now, just to show you that males actually can respond to something, I know Ted said we were going to talk exclusively about females, but for the guys out there, here's males. This is a completely separate stimulus, a cytokine called interleukin-6. The details of that don't really matter, but here's a nice response in the male and a very similar looking response in the female. So it's not as though males can't respond to anything. They do. They just don't respond to prolactin. I'm going to transition now into something that those of you who follow the news and follow FDA approvals and follow drugs for migraine are probably familiar with. But there are a number of new drugs that have just recently been approved in the last few months that are targeted for migraine. They're specifically for migraine. And they're therapeutic antibodies that are targeting this protein called CGRP, calcitonin gene-related peptide. You don't have to write that down. It's CGRP is what everyone says. So there are now 3 drugs, Aimovig, Ajovy, Emgality. I am not endorsing any of those. They're probably all great, but I'm not going to say which one is better than the others. There are now 3. The reason that this was even an idea goes back to measurements that were made on humans. So if you draw blood from the jugular vein of humans who have migraines, during their attacks, whether they have migraines without aura or with aura, there's higher levels of CGRP in their blood during the attacks than if you draw blood and look for CGRP at a time point away from the attacks. So CGRP levels go up in the blood of migraine patients around the time of attacks. And if you treat those humans with triptans, which are the sort of standard acute therapeutics for migraine, that normalizes the CGRP levels. It doesn't go up. So that was a really interesting observation made originally about 20 years ago, led to the interest in CGRP for migraine. The other interesting thing is that if you inject intravenously CGRP into migraine patients, and people sign up for this, this is pain score on the y-axis over time, and look at the pain scores, obviously variable and fluctuating, but clearly an increase in pain scores in the migraine patients when they're given CGRP, but if they're given placebo, nothing really happens, right? So you can measure elevated levels of CGRP in the blood of humans around the time of migraines, and if you give humans, migraine patients, CGRP, you can trigger headaches. That was the original basis that ultimately led to the development of these 3, and there'll be a 4th probably next year. The thing we don't know is exactly where CGRP is doing what CGRP does to contribute to migraine. So we know there's some role for CGRP. That's pretty clear because you can target it and that works in humans, but we don't know where that is. So we asked if we apply CGRP to the meninges, like I showed you earlier, what happens? So here is same kind of data that you saw before. In this case, we're doing this in rats. Previously, it was in mice. Here is blue in females, very nice response. And in males, in red, almost nothing. So that's interesting. That was a pretty high dose. We dropped the dose. We still get a response in females. I'm not showing you the male data, but males don't respond to the high dose. They don't respond to the low dose. Basically, we can put CGRP onto the meninges of females, and we get a nice response. And we do it in males, exact same thing in males. Almost nothing happens. Okay, interesting. Here's IL-6 again. If we give IL-6 to the animals, we get a nice response acutely, we let them recover, and now they're sensitized to an even lower dose of CGRP. We do the exact same thing in the male and focus on this part of the graph here, because when I transition from female to male, you get the nice IL-6 response, but then you don't get the CGRP response, right? So very, very clear difference between the males and the females here. And something CGRP is doing, at least in the meninges, that just doesn't appear to be happening in the males. We looked at the prolactin responses and we looked at the CGRP responses, and they were strikingly similar, that something happens in females and nothing in males. So we thought there may be some crosstalk between the two. here's the response in females to CGRP applied to the meninges. And if you do that in the presence of a blocker of prolactin receptors, you can actually block the response to CGRP by blocking prolactin receptors. So there's some kind of crosstalk between the two systems. And we don't really understand that completely yet. That's something that we're investigating in the laboratory right now. So you may look at this and say, well, there's these 3 antibodies that have been approved, and you're telling me, CGRP is doing something very prominent in females, and it's not maybe doing something in males. Do these antibodies even work in males? That's a great question. We're still trying to sort of get a handle on this. Here's data from 2 different clinical trials in humans of Ajovy, Aimovig. I'm not picking on these 2. The same thing would be true if I had Emgality up here as well. These are the 2 that I chose. But these are the demographics of the patients who were in these trials. And so here's placebo and 2 different doses of Ajovy, placebo and 2 different doses of Aimovig. The thing that I want to draw your attention to is look at the percentage of the patients in these trials that were female. So here's male, here's female. So in the placebo group, 85% of the patients were female. In the low dose Ajovy, 86%, 85, 86%. We can look over here in the Aimovig trial. So what are we at? 86%, 85%, 85% female. It's really actually hard to tell whether these work in males because there's so many females in these trials. Clearly it works in females, and we just don't have a good handle yet on whether it's going to work in males. So to come back to my original question and leave Ted a little bit of time to talk, is there a need for female-specific migraine therapeutics? The short answer to this, I think, is yes. Clearly, migraine happens more often in women, and it's more disabling in women. We need to be going after therapeutics for women. There is clearly some kind of hormonal contribution to migraine, and we just don't really understand exactly what that contribution is. Prolactin may be one of many hormones that's contributing, and it offers a therapeutic opportunity. If we can block the signaling of prolactin, we may have a very good pharmacological therapeutic for migraine that would only work in women, and it probably wouldn't work at all in men. And so that's here. I think we also have to have a better understanding of whether or not drugs work on males or females before we go into clinical trials. The example that I just showed you might have been a lucky example. CGRP therapeutics might work better in females, and it just so happens that migraine is more common in female, and 85% females ended up in those trials, and so the trials looked great. If that weren't the case, and those CGRP antibodies worked in males and not in females, and you enroll 85% females in your trials, you're not going to see an effect, and the trial just failed. So I think we really need to get a better handle on this before we go into clinical trials. And I'll just give you one final example before I stop. This is another hormone that is being investigated for migraine. It's a hormone called oxytocin. And this group did a study where they gave migraine patients a nasal spray, an oxytocin nasal spray, and tried to determine whether or not it worked for their migraines. And so here's some data just in table format. So here's 2 hours after dosing. This is IN or intranasal oxytocin or intranasal placebo. The pain reduction in the oxytocin group, 33% of patients got relief, 26% in the placebo group. That's a pretty high placebo rate, but the actual drug is not much better than placebo in this case. The light sensitivity and the sound sensitivity, 9% versus 21, 2% versus 16, that maybe looks a little bit better, but still not striking. Nausea, maybe a little bit higher. What is the percentage 24 hours later of patients who are rating this as excellent or poor? Not many people are rating this as excellent. A lot of people rating it as poor. It was not reported in the data for this trial what fraction of these patients were male and what fraction was female. If this was 50-50, half male, half female, and maybe this drug only works in females, you're not getting a very good signal here because you have too many males in your trial and it's just not even going to work in males. So don't even enroll males in the trial. So I think this is an example of why we need to get a better idea about sex-specific actions of even drugs that have not been developed to be sex-specific before we put them into clinical trials. So I just want to end with some acknowledgements. This is some of the many wonderful people who work in the lab and do the work. I don't have space here to list everyone that's contributed to everything, but there are a lot of them, and thank all of you. I collaborate with Ted and his lab members, so thank you to them. We're doing all this work on prolactin in collaboration with Armin Akopian, who's down at the Health Science Center in San Antonio, so we're collaborating with him and his group. And then, of course, we can't do any of this work without money. And we've been fortunate to get money from headache foundations, migraine foundations. We've been lucky with money from the NIH, drug companies, and, of course, the University of Texas system. So thank you.
That was fantastic. Thanks, Greg. Appreciate that. So we'll open up the floor for some questions. If there's not one, I actually have one real fast for you.
I planted this with Michael.
Well, actually, it's a different one, sorry. But if you're going to change it up for you, right? So if you experience like photophobia versus phonophobia or aura, does that relate to any part of the particular part of the meninges? In particular, have people found that out?
Yeah, it's a good question. We don't know. We don't know whether there's particular regions of the meninges that are more sensitive, more active. The nerve fibers are more active during migraines. The prominent aura that happens in many patients should be coming from electrical changes in the back of the brain. And so the meninges that overlie the back of the brain should presumably be active, but there's still a lot of controversy about whether the mechanisms that cause aura even contribute to headache. So, I don't think we know the answer to that question. It's a good one, and I would love to be able to answer it, but I don't think we have the details yet.
Yeah, cool. We have a couple minutes if anybody has a question. Absolutely. Give me 1 minute to get to you.
More wanted to confirm that I was understanding what you were saying.
Sure.
For the 3 recent drugs that have been approved and the one that you said is likely next year, are they going to be targeted specifically at females, or are they being targeted to both? In other words, if you were a male suffering from migraines, would your expectations need to be, I guess, tempered about the efficacy of these on your own symptoms because of the drugs? I'm not sure how to word this, because they're working better with women than they are with men.
Well, we don't have the data in humans yet. So I would be a little careful saying that they don't work in males. There are some data that have been released from clinical trials on patients with cluster headache, and those patients that's more common in males. It's sort of interesting. Migraine is more common in females, and cluster headache is more common in males. There are trial data that are now public that these antibodies work in cluster headache, and that's males. It was a higher dose of the antibodies that was given in those trials, so it may be if you take the same dose of the antibody that is working for migraines in females and you happen to be a male, you may have to go to a higher dose. I think all of those things need to be looked at. But to address the first part of your question, those antibodies were not developed to be specifically effective in females. They were developed to be effective in migraine patients. It wasn't really clear, at least until we started looking into this a few years ago, that there is this prominent sex difference with CGRP. And I think now we are trying to encourage the companies to actually ask this question and to power their studies by including more males to really see whether there's a difference. If it's just overall sensitivity, if it's dosage, I think we need to know the answers to those questions. Physicians need to know the answers to those questions because they need to prescribe them correctly.
Greg, thanks. That was great. I guess, doesn't this come down to, in your experimentation, is it just a CGRP thresholding thing that, a very small amount of the CGRP can elicit a migraine in the females versus it won't in the males? But is it clear? Because when you have it, if the males just require a 3-fold greater amount of CGRP to trigger the migraines, anti-CGRP therapies may still have an effect? I mean, am I thinking about this right as far as what your experiments suggest?
Yeah, it's a good question. I don't know whether it's a threshold effect with CGRP yet, because we haven't found a dose of CGRP that actually does anything in the males. And we've gone up pretty high. We're pushing the limits of being able to afford enough CGRP to do these experiments and to get them into solutions. That's not necessarily true with prolactin. I showed you female-specific responses with prolactin. If we go 10 times higher in the dose of prolactin, you can start getting responses in males. They become very unphysiologically relevant at that point because, first of all, it's males, and it's a high level of prolactin anyway, and it's just not going to happen in the male. I think there's a larger question here is, there are lots of triggers that cause migraines in susceptible individuals and don't cause migraines in other people. Stress is the most commonly reported trigger for migraine. I don't get migraines. Does that mean that I'm not stressed? I would argue that's not true at all. I'm stressed all the time, but I don't get migraines. I drink red wine all the time. I don't get migraines. Many people get migraines from red wine. I can go through the perfume section of a department store and never get a migraine, and there are a lot of people who will avoid that location. What is the difference between the people who have migraines triggered by all of those relatively normal things and those that don't? That's a great question. And so, yeah, it might be that the levels of CGRP that cause migraines in females are much lower than they are in males, but why is that? We don't know.
So in the interest of time, let's go ahead and end Greg's section. Thank you. Greg?
Thank you.
And there'll be time to ask questions and follow up afterwards.
So I'm going to introduce Dr. Ted Price. So Ted, here's Ted's talk. Ted, interestingly, graduated from here, this university, University of Texas at Dallas. He was in the first class of bachelor's degrees in neuroscience. So at the time, there were 4 of you.
4 of us, yes.
4. Yeah, that's a 4. One hand. Now there are pushing close to 1,000. So it's been a while, right? I'm not going to say how old Ted is. I'm not going to say what year he graduated, but he was one of 4, and now there's close to 1,000. So he also did his PhD at the University of Texas Health Science Center in San Antonio. We spent a lot of time sitting next to each other there, started working together. Ted went to do a postdoctoral fellowship after he graduated at McGill University, working with an investigator named Fernando Cervero. That's in Montreal. And he was there for several years, and somehow Ted and I convinced the University of Arizona to hire both of us at the same time, and they did. I don't know how that happened, but it did. And so we worked together there for 7 years. We were both faculty members at Arizona. And UTD really liked Ted. They wanted Ted to come back. And they started full court press to get Ted back here. And obviously they were successful. So about 7 years into our time at University of Arizona, we moved here to UT Dallas. Really, they wanted Ted. I didn't graduate from here. They wanted Ted, and I came with Ted. So as I mentioned, we've worked together for a long time. Ted has really done a number of pioneering publications in the field, and there are a lot of areas that we are investigating that other labs are investigating now that first came from him. So he's really been a pioneer in several different areas. We don't really have time to go through all those. I'm sure he can tell you more about them than I can, and he would be happy to do that.
In great detail, yeah.
In great detail, yes. I also mentioned, I don't know do you have a disclosure slide?
I don't.
Ted doesn't have a disclosure slide, but there's a company named after him that ask him about that later, and he'll be happy to tell you about that as well. So, thank you. Ted.
Thanks. Thanks, everybody. All right, so I have a bit of a cold, so I'm sorry I'm going to be sniveling a little bit. And I hurt my knee playing basketball this morning, so I'm also limping a little worse than normal. But okay, so thanks, Greg, for the nice introduction. I'm going to talk about chronic pain kind of more generally. But I want to start by highlighting the great group of people that we work with and the incredible diversity that we have in our laboratory. So this is me, there's Greg. This is the group a couple years ago, actually. I like to put up an old picture because I like to brag on how well many of our students have done. Michael is here, and at that time he was a postdoc, now he's a faculty member, so our really outstanding students do eventually leave and go on to do amazing things. So Michael's doing his own amazing things here at UTD. We also have a lot of undergrads that work in our laboratory, and many of them are pictured here. Two in particular, I want to highlight Khadija, who is a MD student at UT Southwestern. She's just starting her 3rd year, and Sid, who's an MD student at Johns Hopkins, also entering his 3rd year. Many other fantastic students here. I'm going to tell you about work from several people in the lab today, and 2 of them are here. So Candler, who's a PhD student in the lab, not in this picture, this is taken before she joined the lab. It's right over here. I'm going to talk about her work at the end. And Pradipta, who is somewhere right here and also right there. I'm going to talk about his work as well a little bit. All right, so if anybody's heard me talk before, and looking at the faces in the room, many of you have, you know I like this painting a lot. This painting is called A Stab in the Arm, and it's painted by a man named Stan Cohen. And if you were to imagine that he actually was just stabbed in the arm, this is a nice artistic depiction of the neuroanatomy of the pain he feels from that stabbing. So these wirely looking red things here would be the sensory neurons called nociceptors that innervate our body. Those allow us to detect harmful or potentially harmful stimuli and get away from them. Okay, so that number one is really important. If something is causing you pain, you need to be able to get away from it. So nociceptors, these sensory neurons that innervate your body, they serve a vital protective function in that regard. And they also tell you to stay away from hurting yourself again if you're healing. So the reason I can barely put any weight on my knee right now is because the nociceptors innervating my knee have detected that I have injured my knee, and they're trying to tell me, stop hurting your knee, stop walking around on your knee. And I keep walking around on my knee. So they serve a vital protective function, not only to tell us to get away from things that are hurting us, but to protect ourselves while we run the healing process. So unfortunately, that's not really what happened to Mr. Cohen. What happened to Mr. Cohen is that he had a cervical spinal cord injury. He had a vertebral disc that blew out, and it caused him to feel, when it happened, like he was being stabbed in the arm. So unfortunately, he continued to feel like he was being stabbed in the arm. He had a surgery to fix the busted vertebrae, and even though he had that fixed, and it no longer appears, if you do an MRI, his vertebrae seem to be normal, he still feels like he's being stabbed in the arm all the time. And this is really and it's 10 years after, okay? So this is really the problem that people have with chronic pain. It's that they have some kind of injury. Migraine, which Greg just talked about, is weird. We don't really understand it very well because there doesn't seem to be any injury. It just emerges and then doesn't go away. Many other types of chronic pain, people will have some kind of injury, and then they either have continuous pain even though the injury resolves, or their threshold for pain drops dramatically such that they'll have episodes of pain. So low back pain is a really good example. Many people that have low back pain will have waxing and waning of the low back pain. They may have some really minor injury that ordinarily wouldn't bother them, or they may just bend over to pick up their kid's ball in the kitchen and suddenly can't walk, right? So it has been viewed for the longest time that pain is just a symptom of another disease. But I think what we now know from the last 10 to 20 years of work is that that's really not true. That chronic pain is a disease in and of itself, and it's a disease of the nervous system. So we strive very hard to try to understand that, and I think it's really important because it tells you about how you should try to treat chronic pain. And it's not a symptom in most cases of an underlying disease. It's a disease in and of itself, and what we really want to try to do is reverse the plasticity in the nervous system that's causing it. So there's really 2 big things happening right now in the pain world. One is that we have a huge opioid epidemic that everybody has heard of, and that is depicted here. Here you can see the number of people that are dying of opioid overdoses every year from 1980 until currently, and this is undergoing exponential growth. Hopefully that exponential growth will quickly start to curtail, but there's been an absolute explosion in this, and it's not just in heroin, but also in prescription opioids. In fact, the growth of addiction and overdoses to prescription opioids is fairly remarkable. And many of these drugs that people are dying from, like fentanyl and carfentanyl, are actually quite dangerous drugs that really were never intended to be used outside of the hospital. So the point of this lecture is not to talk about opioids, but it's hard to talk about the problem of chronic pain without talking about opioids. So to solve this epidemic, I would argue that we need to have a better ways to treat addiction, number one. Number two, naloxone, which is an antidote to opioid overdose, is widely available, and our politicians are just not making sure it gets into the hands of people that need it. So if we had the political will to do that, we could save thousands of lives. So that's something that you can advocate for actively, to say, you know, naloxone should be available at universities like UTD. So at many places, they're putting them in the cardiac electrical, I'm forgetting the name of it right now, but they have that in case somebody has a heart attack, and also naloxone in case somebody has an opioid overdose. It saves people's lives 100% of the time. So this is one major issue. And then the third way that we can combat this is by coming up with better ways to treat pain. So there's also a chronic pain epidemic happening in the United States right now. We estimate that about a third of the population, 100 million American adults, suffer from chronic pain. That's defined as pain lasting for longer than 3 to 6 months. It costs a staggering amount of money. This is greater than the cost of cancer, diabetes, and heart disease combined. So chronic pain is a huge, huge burden on the healthcare system. It disproportionately affects the so-called socialized part of the healthcare system, so Medicare, Medicaid, and the VA system. So there's a huge amount of money that is spent on that. And obviously, it dramatically reduces quality of life, as anybody who suffers from chronic pain or from migraine knows. So we need better ways to treat this, and I would argue that we need ways to treat chronic pain that are non-opioid, which is really what both of our labs is focused on. You often hear people say that, okay, well, you know, this is a major healthcare problem, but it doesn't kill people. Well, that's also not true. These are statistics published very recently in Annals of Internal Medicine, looking at people that commit suicide. So up to 10% and increasing of people that commit suicide are thought to commit suicide because they have chronic pain. So they're choosing to end their life rather than continue to deal with the pain, the chronic pain that they're unable to treat. And most of these people end up killing themselves by shooting themselves. They don't overdose on opioids, which is another idea that people have, that people who have chronic pain are dying of opioid overdoses. That doesn't seem to be the case. So the point of the discussion, now that I have everybody thoroughly depressed, is to talk about how we can potentially better treat chronic pain. And what I want to focus on today is the idea that we might think about treating chronic pain differently in men versus women. So up until about 5 years ago, almost all of the work that was done in academic labs on pain and most other areas of neuroscience was done entirely on male animals, usually rodents, or in male subjects. About 5 years ago, the National Institute of Health said that we're no longer going to fund research that only looks at males, and you have to consider looking at males and females in your work, and basically now it's impossible to get funded if you're just going to study males in your work. On top of that, these are some additional statistics from the NIH showing you how many women scientists get predoctoral and postdoctoral fellowships, and then go on to also lead major research projects. And we're also failing miserably here. So you can see that at the start of women's scientific careers, They're getting fellowships at about exactly the same rate as men are. But by the time they advance into their careers where they would be leading big research projects, between 70 and 80% of these are led by men and not by women. So I would argue that this is also a failure, and it's a big reason, I think, why so much research has historically been done on male animals, is because the work is being done by men, and it's a big part of their bias. So these numbers are actually starting to get better, but they've been very poor for far too long. So I'm going to talk now about our work in pain research on sex differences. So also historically in this field, most of the work has been on this idea that men and women may perceive pain differently, or men and women may have different pain thresholds. That's not what I'm here to talk to you about today. This has been studied ad nauseum, and the effect sizes are very small. Basically, men and women perceive pain in about the same way, and they have about the same pain thresholds. What is quite different is what Greg talked about before, and that's that chronic pain diseases disproportionately affect women. So why? And what are the underlying mechanisms? That's really what we're trying to understand. So when Michael, who's right here, joined our lab, we mostly weren't looking at the way the immune system interacts with the nervous system, but we now pay a lot of attention to that, and it's led to some really, I think, remarkable findings about how mechanistic differences in generation of chronic pain occur. So over the last 10 years, we've really gained an amazing understanding of how these nociceptors that I talked about at the beginning of the talk interact with the immune system. Before people started doing this kind of work, we essentially thought that the immune system kind of acted on its own, the nervous system did its own thing, and the two of them didn't interact at all. We could not possibly have been more wrong about that idea. What we know now is that peripheral and central immune cells like macrophages, mast cells, and other types of immune cells like T cells release all kinds of factors, and almost every single one of those factors is able to act on sensory neurons to sensitize them. But what we also know is that this seems to happen in a much different way in males and females. So I'm going to give you one example of some work that we've done in our lab connected to this. So we were working with a very talented chemist at Duquesne University named Yelena Yannick, who developed what she calls Therapeutics. So basically, she takes a molecule and she encapsulates it in what she calls a nanoemulsion. And what that can do is protect the molecule so that it doesn't get metabolized when you take it, okay? So it gives the molecule a longer half-life, and it also can target it to specific types of immune cells. So we knew that macrophages seemed to play a really important role in promoting pain after injury. So we thought, you know, we can take this molecule that has a powerful biological action, which is resveratrol. We can package it in this nanoemulsion and prolong its half-life, and also target it to macrophages, because macrophages basically like to eat this nanoemulsion. And wouldn't that be a cool therapeutic? So what we did was to do a very simple model. It was a post-surgical pain model. We do an incision on a mouse paw. We give it this nanoemulsion, and we measure the amount of pain that they have. So Greg already told you about how this works. The animals become very hypersensitive. In the male animals, we give them the resveratrol nanoemulsion, and it pretty strongly inhibits their incision-evoked pain. So that was expected because there's all this literature on macrophages promoting pain after injury. So what do we see in the females? Absolutely nothing. Had no effect whatsoever. I guess I also was wrong. We are going to talk about stuff that's for the guys, right? All right, so this thing could work for men. It doesn't seem to work at all for females. So we were scratching our heads like, what on earth is going on here? So we did a kind of crazy experiment where we looked at the nerve that innervates the hindpaw in the mice, and we asked whether or not it was filling up with macrophages. So what this shows on the ipsilateral side, so the side on which the mouse had the injury, this is the nerve. The green things are all macrophages. The nerve is just completely filling up with macrophages. On the other side, this is the epineurium. This is the outside of the nerve. Here's the nerve itself. You can see there's essentially no macrophages. So in the male animals, the resveratrol nanoemulsion works like gangbusters, and the nerve from this really little injury is completely filling up with macrophages. So what happens in the females? basically nothing. There's a little bit of macrophage infiltration in the nerve, but it's pretty small, and it's not any different than what you see on the contralateral side. And if you quantify this, you can see in the males, there's a really big increase in macrophage infiltration into the nerve. It lasts for about 2 days. And in females, basically nothing happens. So the therapeutic is very specific. It works only in males. And the underlying biology is completely different between the males and the females. So we also know that this is consistent, that there's a type of macrophage found in the central nervous system called microglia. And we also know that these microglia seem to play a key role in promoting pain in male, but not female rodents. So we now want to try to understand what's actually happening in humans. So we've started this big project with MD Anderson in Houston. What we're doing is that they're bringing in patients that are having a spine surgery. So that they have cancer that's at MD Anderson Cancer Center. They have cancer that's infiltrated their vertebrae, and they need to have their spine stabilized because they're at risk for spinal cord injury from the degeneration of their disc from the infiltrating cancer. What that gives us the opportunity to do is something that we ordinarily can never do in live humans, and that's obtain some of the nervous system tissue. So when they put these metal rods in to stabilize their spine, the dorsal root ganglia, which is where these nociceptors are in the peripheral nervous system, come out as part of the surgery. And what we do is we use this advanced sequencing technique called RNA sequencing to look at what's changing in people that do or don't have pain, and looking in males and females to see if it's different. So let me just give you a little taste of what we found. And this has been what Pradipta has been working on for some time in the laboratory. So the genes here are not really that important. I want you to pay attention to the colors, though. The yellow represents the genes that are increased in the males. The purple shows the genes that are increased in the females. And if it's got a little green box on it, it's a macrophage gene, okay? So first of all, hopefully you can appreciate of the genes that are changing, many of them are in macrophages, right? So in humans that have this kind of pain from problems with their spinal cord, they have a lot of macrophage infiltration that presumably is driving the pain. But if you look at where the yellow boxes are versus the purple boxes, hopefully you can easily appreciate that the male is skewed very strongly in this direction, and the female is skewed very strongly in the other direction. So what is over here? Well, basically this is the classic cytokine response that we all know about in the literature. Activation of cytokines and chemokines, and it happens almost exclusively in the males. In the females, we actually see something very different. We don't see all these cytokines getting turned on, and we see a lot of genes that we know are regulated by a particular factor called nerve growth factor that is increased in macrophages, specifically in this case in females. And that likely is driving expression of a bunch of new genes in the nociceptors in females. So the mechanisms of plasticity that are driving pain in these human patients seem quite different between the men and the women. So what can we ultimately do about this? Well, I think we found something else, and this is Candler's work, that's pretty exciting in this regard, and that is CGRP receptor antagonists. So Greg talked about this before. CGRP is all the rage in the migraine field right now. So is it possible that all of those drugs that were just approved for migraine could actually be used to treat all kinds of pain in women? So this is kind of what Candler is testing here. So we're doing a model we call hyperalgesic priming, but the model is not really important. What's important is the effects of the CGRP receptor antagonists. So the female is on this side, the male is on this side. If we give the injury and give the CGRP receptor antagonist to the females at the time of injury, it blocks the injury-induced pain response and blocks the development of chronic pain. And even when chronic pain is developed, if we block CGRP receptor antagonists in females, we can reverse the chronic pain, whereas in males, it does nothing, okay? So there's no effect here, no effect here. So CGRP obviously plays a really important role in promoting pain in migraine, but it may also play a key role in promoting pain in females in a broad variety of conditions, and that may just not be the case in males. So you asked earlier about whether or not this is a difference of degree. In the spinal cord system, it is a difference of degree. So the curve here is just shifted way to the left for CGRP. So females are just way more responsive to CGRP than are the males. It's not like the males don't respond to CGRP, but they're probably only responding to concentrations of CGRP that are outside of the physiological range. So again, I did choose only one of them. This was the first one that got approved. There's this possibility that, this drug, these 3 drugs that were just approved for migraine may actually be effective for a variety of different types of chronic pain in women. So we'll see if this can ultimately be tried. But once a drug is approved, it can be used for off-label clinical trials sponsored by individual investigators. So this is a question that can actually be addressed now. So I want to just quickly wrap up by saying that we've started this new Center for Advanced Pain Studies at UTD. We have a bunch of really great faculty that are involved in this center. I think we're doing some pretty exciting things, and we're also extraordinarily interdisciplinary. So we have investigators from behavioral and brain sciences who sponsored this event, but also from many other schools, including natural sciences and mathematics, engineering, computer science, also from the business school, which we're in right now, and we have affiliate members from UT Southwestern. And I just want to again thank the really fantastic people that work in my lab, many of whom are here, and who you can talk to about their work after any questions. Thank you.
Fantastic. That was fantastic, Ted. Appreciate
Thank you,
that.
Michael.
So does anybody have any burning questions? If not, I actually have one.
Hopefully it's the one we planted.
I don't think, it's still not the one you planted, sorry. You come up with different things, it's a question that I often am asked to think about a lot is the onset of sex differences is apparently more and more real, but what kind of explanation can there be evolutionarily for those differences?
So that's a question that I've gotten many times, and I have to say I don't have a very good explanation.
Okay.
I think that the idea in the field has been, this is not really a direct answer to the question, but it's the way I'm trying to think about it now. So the idea in the field for the longest time has been that sex hormones that really start to raise their levels at about the time of puberty onset are responsible for these sex differences. But there have been a couple of papers that were published very recently that I think turned that idea on its head and demonstrate that if you look at the immune system specifically, very early during development, that there's strong sexual dimorphisms that are found basically from the start of the birth of the immune system that last all the way into adulthood, and the influence of sex hormones is actually relatively small. So for instance, if you take female animals and look at the transcriptome of their microglia at the time of birth, and also take animals that you overectomize so they don't produce any estrogen, and look at the phenotype of those cells late in life, the influence of estrogen is actually very small. So I think that we probably need to think about how we look at this a little bit differently, and that sex hormones, you said when you were talking about migraine that it's actually confusing, right? That it's not clear at all that sex hormones play a clear. They're obviously involved in what's going on, but they're involved in the basic underlying biology. And one of the reasons that very few people did sex differences research before NIH told us we had to is because everybody was scared of trying to control for the differences in hormone levels during the estrous cycle in female animals. And so once we all started to do this work, nobody really was controlling for that. And what do we find? That it really doesn't make that big of a difference. It doesn't matter what stage of the estrous cycle they're at. You see these marked sexual dimorphisms independently of changes in sex hormones.
Absolutely. Absolutely.
So I totally avoided your question, but.
Oh, you got
yes.
it.
So this is kind of for both of you guys. we've looked at sex differences, but what about differences in different races and like Caucasians versus African Americans versus Asians? Are we moving towards looking at that? Because we know like different races have different enzymes and kind of work differently, but is that kind of where
So-
things are going?
Well, we can't do that in animals. We do, the work that I have seen in the pain field has looked predominantly at disparities in access to healthcare. And pain outcomes are much worse among African Americans and Latinos than they are among Caucasians. And the reason for that, as these studies have shown, is that access to healthcare is poor. So I think that actually is the most important thing for us to be working on, is solving the issues of these disparities, because it's driving what appear to be differences that people may think are driven by underlying biology, but in fact are actually driven by disparities in our society.
You want me to go after that too? So I might actually answer that a little bit differently, but so in the case of migraine, it's most, if you look across races, it's more common in Caucasians, a little bit less common in Asians, and then the least common in Africans. So I mean, there is a stratification. We don't really understand why that is. Is there a genetic basis to it? People ask that question all the time. Is there a genetic basis to migraine? And we can expand this. Is there a genetic basis to pain? In very limited cases, yes. But for most migraine patients, no. There have been a whole lot of genetic analyses that have been done now on large numbers of migraine patients. And it just does not appear to be at least a single gene. It might be a polygenomic disorder. But migraine is clearly familial. If you have one parent that has migraine, you're 40% more likely to get migraines. If you have two parents that have migraine, you're 90% more likely to get migraine. That doesn't have to be in the genome. Many migraine patients have attacks that are triggered by specific dietary habits. Well, how did you learn your dietary habits? You learned them from your parents. You maybe are carrying on patterns that you learned when you were young. And so I think, it's really hard to address that. There might be, environmental issues that contribute to whether or not you become a chronic pain patient that really have nothing to do with your genome. And it might be something that you did when you were very young, or someone did to you when you were very young. And those are very hard things to look at, but I think they're important. So, across societies, across races, people behave differently, people eat differently, people do different things. All of that might actually contribute to the prevalence of pain in those populations, and it just is something that we need to examine.
Great. So I think that is pretty much it for time. So thanks to you both.
Thank you. Thank you all. Thank you all for coming.